Hello and welcome, everyone. It's my pleasure to introduce today's speaker, Dr. Ted Cohen, who is a professor of epidemiology at the Yale School of Public Health with a research focus in tuberculosis. He is particularly interested in understanding how drug resistance and medical comorbidities such as HIV frustrate current efforts to control tuberculosis epidemics with the goal of developing more effective approaches to limit morbidity. We're very excited to have you here today, and the floor is all yours. Thank you.
Thank you. It's great to be here, great to be back. I did my training here. Mark was one of my dissertation advisors, and I trained with Megan Murray, who's now at the med school. I want to talk about sort of an arc of work that is still ongoing in the Republic of Moldova. I did want to point out that this work was funded by USAID and NIH. I normally wouldn't point that out transparently, but I think in this moment, sort of recognizing the funding necessary to do the type of work I'm going to talk about today is quite relevant. So I know some of you know quite a bit about tuberculosis. I'm not going to sort of dwell on a lot of the details of the biology or natural history of this pathogen, but I do think to get everyone up to speed, I wanted to talk a little bit about both the natural history and burden of this infectious disease. So tuberculosis is caused by a bacteria called Mycobacterium tuberculosis. It has a complicated natural history. Most of the individuals that are infected with TB never progress to disease and are in what we call sort of a latently infected state, during which they're not experiencing any symptoms, they're not infectious to any others. And then a minority of individuals infected can progress to infectious tuberculosis. And this is characterized by a number of different potential symptoms, but individuals with TB in their lungs or pulmonary TB may be infectious to others. In that stage, disease is diagnosed either by seeing the bacteria and sputum through microscopy or by growing up the pathogen, tuberculosis, Mycobacterium tuberculosis, in culture, or more recently through the detection of the genetic sequence of TB through a number of different means. This graph on the left represents the best estimates that we have of the global burden of tuberculosis. So this is a pathogen, despite the fact that it is curable with antibiotics, that causes the most number of deaths due to an infectious pathogen in the world. We have approximately 10 million cases a year and approximately 1.25 million deaths due to this infection. The work I'm going to be talking about today is focusing on drug-resistant forms of tuberculosis. Here I've sort of put up these categories of MDR or RR tuberculosis. This stands for either multi-drug resistant or rifampin-resistant TB. Multi-drug resistance is defined by resistance to both rifampin and isoniazid, two of the most important first-line drugs to treat TB. And because of some of the new diagnostics which just detect rifampin resistance through molecular means, these have been generally grouped together in reporting most recently. This particular phenotype, either rifampin resistant or multi-drug resistance, is particularly important from a clinical standpoint because these individuals require longer treatment regimens, typically with more toxic drugs. But encouragingly, we have some new drug regimens that look really promising and are now being scaled up in the field. The graph on the left in the green, what you're seeing is the estimated global incidence of multi-drug or rifampin-resistant TB. So about 400,000 cases of this highly resistant TB are estimated to happen every year. The lines below that represent the number of diagnosed and detected cases and those that are placed on appropriate treatment. You can see that we're probably only detecting and treating about half of these individuals with highly resistant disease. So that's a massive failure and we have concerns about what this means for the ongoing transmission of TB and drug-resistant TB, which I'll talk about in the rest of this talk. I do want to point out on the right some global estimates of the fraction of TB cases that are drug-resistant. And you see there's two lines here. The top one refers to individuals who have had no prior history of treatment with TB. And so on a global scale, about 3 to 4% of individuals who have never before been exposed to TB drugs have drug-resistant disease. So these individuals were certainly infected by a strain that was already resistant to these important first-line drugs. Then there's a smaller group of individuals who have been previously treated for TB. So these are individuals who have already experienced treatment in the past and are now returning for treatment again. As you can imagine, these individuals are enriched for having drug-resistant disease because they may have been treated for drug-susceptible disease in the past, acquired drug resistance, or maybe had been infected by a drug-resistant strain, treated with an inappropriate regimen, and now requiring treatment again. And the global level of drug resistance in these individuals is substantially higher. But what I'm going to emphasize is these are global estimates, and these can mask really important underlying heterogeneities. This map shows us the estimated incidence of multi-drug resistance at a country level, and what you'll note here is that the largest circles representing the biggest burden of multi-drug resistant tuberculosis are in India and China. This is driven largely by sort of the huge population numbers and the relatively high incidence of TB in these countries. Turns out that a relatively small fraction of individuals in these countries have multi-drug resistant disease, but the sheer number of cases means that we also have a lot of drug resistant cases in these regions. I want to contrast that with what we see in the Russian Federation and in Eastern Europe, where although the overall burden is smaller, the risk of drug-resistant TB among those that have TB is much higher, and we'll see that in the Moldovan data. So I want to turn to Moldova. This is a relatively small country in a really geopolitically vulnerable setting. So Moldova resides between Ukraine and Romania. It's a landlocked country of about 3 million individuals. It gained its independence from the Soviet Union as it dissolved in the early 1990s. There's a really important region, which we'll talk about here, which is in this eastern region. There's a river that separates it from the rest of Ukraine. This is a semi-autonomous region called Transnistria. It's not internationally recognized. It's really part of Moldova from an international standpoint, but it operates sort of semi-autonomously. It's a Russian loyalist region. There's Russian soldiers that monitor the borders there, separate postal system. So there's some really important differences between what's happening in Transnistria and the rest of Moldova. The estimated incidence of TB in Moldova is about 84 cases per 100,000 individuals. This is a high burden of TB, but it's not remarkable. What's most remarkable about it is the fraction of cases that are drug resistant. So given their burden of TB, the level of drug resistance is exceptionally high. Estimated about 1 in 3 new cases. These are cases who have never before been exposed to TB drugs, and more than half of retreatment cases have multi-drug resistant disease. So why study TB in Moldova? So from my perspective, Moldova is an opportunity not only to study disease within this very important country from the perspective of drug resistance, but also it provides a window into what's happening in the larger region in Eastern Europe and Russia. This work has been made possible through sort of the hospitality and generosity of my colleagues in Moldova. I think some of them may be joining this meeting. um, by, by Zoom. I want to call out in particular Professor Valeru Crudu, who is the head of the National Reference Laboratory in Moldova, and his colleagues Nelly Ciobanu and Alexandru Codreanu, who work in the lab and have been close collaborators since we started working there in about 2010. There are some particularly remarkable things about the TB system that they have developed in Moldova that have allowed us to do the type of work that we've been doing there, and I want to call those out. In particular, they have invested heavily in their ability to do microscopy and culture for all individuals suspected to have TB, and now also using GeneXpert, which is a molecular tool to detect both TB and rifampin resistance at the time of diagnosis. Also, they have universal drug susceptibility testing for all culture-positive TB cases, and they also follow individuals on treatment to ensure that they're getting appropriate treatment. And for individuals that remain culture-positive, despite being on treatment, they do repeat drug susceptibility testing to get these individuals onto the best possible therapies. Valeru also invested in an electronic TB database. We may take this for granted. This is remarkably different than other countries in the region. So the ability to actually dive into the data and help with analysis was made possible through Valeru's insight in developing this system more than 15 years ago. So I'm going to first talk about work done by Helen Jenkins, who's now faculty at BU. She was a postdoc with me here at Brigham when she did this work. So we were invited in by Valeru and his team to help them analyze data that had been collected routinely through this electronic database. So in total, what these maps represent is data from more than 23,000 cases of TB occurring between 2007 and 2010. And to orient you here, so in these columns, we're looking at maps of resistance among never before treated cases. And what we're showing on the top here, these colors represent sort of the hotter you get, the higher the risk of multi-drug resistance among cases. So in some places, we see the risk of multi-drug resistance among cases greater than 70%. Here, we're estimating the incidence of multi-drug resistance. So these maps don't totally line up because population differs by location within the country. And these are similar maps for individuals who have been previously treated. As one might expect, both the risk and the burden are different for previously treated cases. But one of the things that jumped out at us was sort of this concern of these hot spots where the vast majority of individuals never before treated for tuberculosis already had drug-resistant disease, really heralding the fact that we're probably seeing a lot of transmission of multi-drug resistance in this region, and a higher amount in some localities than others. Sort of further emphasizing that point, sort of very simple analyses of these routinely collected data also point to not only huge risk of transmitted multi-drug resistance, but also the possibility that this risk is increasing over time. So what we're seeing here are data divided by the age of the patient at the time of diagnosis, and the height of the bars represents the risk of multi-drug resistance among those cases. And they're separated into new cases, previously treated cases, and then the weighted average of those two. And what you can notice, even without any analysis, is that there's this general downward trend. So younger individuals with TB are more likely to be drug resistant, suggesting that these more recently circulating strains are more likely to be multi-drug resistant. And that bears out in multivariable models where we try to adjust for other covariates, where we find that indeed younger age is associated with greater risk of multi-drug resistance. So on this foundation of working together on already collected data, we began to sort of branch out and do some prospective work. And the first study that we did together was based on a single municipal hospital in the capital city of Chisinau, where we enrolled, or attempted to enroll, all culture-positive TB patients admitted over just over a year period between October 2013 and December 2014. And in total, we were able to collect initial diagnostic isolates. So these are isolates before initiating therapy from 283 patients. And this is the phylogeny that was generated from these data. I do want to call out Tyler Brown, who is now faculty at BU as well, and Barun Mathima, who's at Columbia. Many of you might know Tyler. He spent time here at CCBD. There's a few things that I want to call out here. So on the left, we have these MTB lineages with some numbers in front of them. So we have some representation of lineage 2.2, here shown in red. That's represented in this part of the phylogeny. And then we have a bunch of sublineages of lineage 4. We're going to come back to this repeatedly. But what I want to highlight here, so along these columns, we have flagged mutations in the genomes associated with resistance to important drugs used against TB. On these columns, we have resistance mutations associated with isoniazid resistance. In these columns here, we have resistance mutations associated with rifampin resistant. So those that have blue bars in this column and this column down here are multi-drug resistant. And what's clear is that all of the multi-drug resistance is located in either this lineage 2 background or in this densely populated lineage 4.2 background. And this was eye-opening for us. This lineage 2 background is called the Beijing lineage. It has been widely associated with the spread of multi-drug resistance in Asia and the former Soviet Union. This finding of a large, highly clonal group of MDR strains on this lineage 4.2 background had not been reported before, and was something that we were quite surprised about in seeing these data. So, to give a little context here, here's the MTB complex. It's composed of 9 lineages descended from M. conneti. All large transmitted multi-drug resistance epidemics have been attributed to these lineage 2 strains. That's part of the East Asian lineage, and lineage 4 strains, the Euro-American lineage. But in the region that we're looking, all of the previous data suggest that drug resistance and the transmission of drug resistance is associated with these lineage 2 strains, not these lineage 4 strains. And in particular, there has been many publications and a lot of concern about the rise of what is called the W lineage. So for those of you who may remember, we had some outbreaks, nosocomial-associated MDR TB outbreaks in New York hospitals. That was due to a close relative of these W strains. And there's been some really interesting work collecting strains from this region and estimating that the real sort of population expansion of this particular lineage 2 family of MDR strains coincided with the collapse of the Soviet Union. So we asked, are there any data to help us understand whether what we're seeing just in this capital city of Chisinau may reflect a larger problem in the region? And so there was there still is a database that tries to collect some TB whole genome sequence data. And in this work with Tyler and Barun, we attempted to compare what we were finding in Chisinau with other strains that had been sequenced in the region. And so on the outer parts of this tree, you can see the lineage numbers. We have lineage 4.2.1. This is our the lineage that we were particularly concerned about. We have the lineage 2 strains, the so-called Beijing lineages. Just to give you a sense of what to focus on here, the nodes here are colored by the country of origin of the patients that these samples were obtained from. And on these outer bars here, I'd like you to focus on the red dots, the red dashes are strains that were multi-drug resistant. And the blue bars are strains from the study that I'm currently talking about in Moldova. So a couple things to appreciate are this particular highly drug-resistant lineage 4 strain, entirely MDR, and almost all these strains were collected in the course of our study in Chisinau. The other strains that were in the collection were also collected from Moldova. There's a few from Romania in there, a couple from Russia, but all of these data seem to originate from Moldova for the most part. So all of this was concerning to us, but still, this is sort of insights generated by a single study done at one period in the capital city. And so we sought support to do a countrywide whole genome sequence study, and we were able to enroll patients prospectively for 2 years, so from the beginning of 2018 to the end of 2019, enrolling all TB cases and individuals that were culture positive. We collected their MTB isolate, did all of the phenotyping, and did whole genome sequencing. As expected, more than a third of cases were multi-drug resistant, a higher percentage in retreatment cases than new cases. This is the phylogeny that was generated by this study. I want to point out Changwang Yang, who's now faculty at Sun Yat-sen University in China, and Ben Sobkowiak, who is a bioinformaticist who we've been working with for the past several years. This is their work. And we saw, as in the previous study, but now looking countrywide, that multi-drug resistance was concentrated in 2 sublineages. The Beijing sublineage, which has been reported widely, as I mentioned, of which we had more than 900 strains. Nearly half of them were multi-drug resistant. But the sublineage of lineage 4, what is called the Ural lineage, 4.2.1, of which we had more than 500 cases, 3/5 of these cases were multi-drug resistant. You can see that in this figure here. So we've unrolled the circular tree here. You can see lineage 4 strains are here in the blue. Lineage 2 strains are here in the red. Here, these first 2 rows represent mutations associated with drug resistance to rifampin and isoniazid. And you can see the concentration of multi-drug resistance in this Ural lineage, and then sort of a less concentrated, but still highly prevalent phenotype of multi-drug resistance on this Beijing background. So to give you a sense of how we tried to dive into the transmission dynamics and trying to understand the spread of these strains in Moldova, I wanted to first provide some maps where we tried to look at the estimated notification rate of culture-positive TB in the country by locality. And you can see there's no really sort of strong patterns of areas where we see higher incidence than others. Of course, there's heterogeneity. Here are the locations of all of the cases in our study of lineage 4 and lineage 2. So lineage 4 is here in blue and lineage 2 is in red. Difficult to pull out patterns from this. Some of our work sort of trying to extend from these phylogenetic trees to inference around transmission relies on tools that are developed by one of our colleagues who also did her postdoc here at HSPH, Carolyn Coline. I won't talk deeply about this method, but many of you are familiar with TransPhylo, which uses a model that incorporates information about within-host generation of variants and transmission to make inference going from a tree like this to a transmission network where we try to understand how individuals that were sampled might be related to one another through transmission, allowing that sampling in these epidemics is certainly partial. We're missing cases for any number of reasons. Based on this analysis, we estimated that 84% of all of the samples collected over these 2 years were in putative transmission networks, and 92% of the multi-drug resistant strains in our sample were part of putative transmission networks. So what does this look like? So here we have these labels of these transmission networks are arbitrary. They're just sort of in order of how we identified these. They don't mean anything. The height of the bars represents the number of cases within that particular transmission network. The color of the bars represents whether that particular transmission network is one that belongs to lineage 4 or lineage 2. I want to point out a few things. The first is that the majority of the cases in Moldova over these 2 years were part of 35 what we call large transmission networks. So these were networks that included at least 10 cases. The second point is sort of related to the coloring within these bars. So solid bars, the fraction of the bar that's solid represents the fraction of isolates within that transmission network that were multi-drug resistant. So for example, these two largest transmission networks were entirely multi-drug resistant. The first one of lineage 4 and the second one of lineage 2. There are some networks that I'll point out that are a mixture of MDR and non-MDR strains. Okay? The other thing I want to point out, which I'm going to talk about on the next slide, are these astraces. These represent transmission networks where we found statistically significant evidence of local concentration of these strains, so spatial aggregation of these strains. And this is work done by Yulan, who's a postdoc in my group whose background is in geography. And she, along with a biostatistician who I'll mention at the end of the talk, named Josh Warren, developed a Bayesian hierarchical multinomial logistic regression model to try to identify specific localities within Moldova that have sort of an overrepresentation of any of these particular transmission network lineages, respecting the fact that we expect that there's some spatial correlation of this. And so what we're seeing here are sort of these arbitrary transmission networks. These represent the 5 networks where we did find some localities with localized transmission. And you can see that 4 of these are due to lineage 2 strains, and 1 of them is due to a lineage 4 strain. What I'd like to point out here, and sort of I'm reproducing the Beijing map for this transmission network 75 and this transmission network 178. So these represent our 2 largest transmission networks, one which is this Ural strain, and one which is this Beijing strain. What is perplexing to us is here we have a widely geographically distributed, highly drug-resistant strain that we haven't seen reported in any other places, whereas our Beijing strains seem to be fairly locally transmitted. Perhaps some of that has to do with, and maybe not perhaps, I think it's reasonable to suggest that sort of the concentration in Transnistria may represent sort of limitations of population movement from one area to another. So we had real questions about sort of what might be underlying the apparently different transmission patterns of these Ural MDRs and Beijing MDRs. This is work done by Melanie Chitwood, who just defended her dissertation and is doing a postdoc with us. She did her master's here. I'm emphasizing this because so many of the people in this field have had training here. And being back here reminds me sort of what an opportunity it is for you guys to study and work here. So I think that's inspiring. But what I do want to point out are, so we're reproducing the data from the Ural strains and the Beijing strains. Melanie used several different methods to try to find some structure in these trees. These are clades that we've labeled based on these time phylogenies. Some things to note are we see that the MDR phenotype is concentrated entirely with what we're calling this Ural A clade, whereas MDR is sort of more distributed within these different Beijing clades, although some are more completely MDR than others. So Melanie, in her doctoral work, was really interested in trying to understand if we could tease out reproductive fitness differences between these strains. Are these Ural multi-drug-resistant strains behaving differently than Beijing multi-drug-resistant strains in terms of their ability to transmit and cause disease? And this is challenging to do, and she used several different methods to try to get at aspects of this. So the first method she used was to calculate these local branching indices, which are basically using information about the shape and branching patterns of the trees. Sort of the underlying sort of heuristic is that sort of the flatter the tree, sort of the more comb-like it is, the more closely related progeny, the more fitness we assign to a strain. So strains with higher local branching indices may be associated with greater reproductive fitness. Ooh, my colors importantly got lost here. Okay. All right. This is going to, okay. So in the interest of communicating this as best I can, I'm going to just focus on this left-hand panel here. I'm worried about some of the next figures too. But okay. So what I'd like you to compare here, here we have phenotypically non-MDR cases and cases that were multi-drug resistant. You'll have to believe me that this particular hump, this one, represents Beijing lineage strains that are not multi-drug resistant. And this is Beijing lineage strains that are multi-drug resistant. The point being that as these strains acquire or associate with multi-drug resistance, we don't see big changes in our estimate of the local branching index. And this is consistent with what other people have found experimentally, is that Beijing lineage strains seem to be able to acquire the mutations associated with resistance without major losses in fitness. So that is not new, but that is recapitulated in our data. What is more concerning and interesting here is that if you look at the local branching index for the Ural strains, we see a higher local branching index for the multi-drug resistant strains compared to the non-multi-drug resistant strains, suggesting the possibility that these multi-drug resistant strains are spreading more quickly than their closest non-drug resistant relatives. A second approach for doing this, also based on the phylogenetic trees is tried to estimate she tried to estimate the population sizes of these different clades over time. The underlying approach here is to use the shape of these trees to correlate with sort of population growth patterns, where populations that are growing exponentially in time, we'd expect to have these longer external branches compared to internal branches, and populations that are constant over time, we'd expect to have relatively short external branches compared to longer internal branches. And when she did this for all of the 9 different clades, 3 of the Urals and 6 of the Beijings, what was remarkable is sort of this one clade, our Ural A clade. This is the clade that is almost entirely MDR, seems to be growing exponentially, while the other clades are sort of bopping along with some indication of changes over time, but quite a different pattern estimated to be happening for these Ural A's. And finally, Melanie used a birth-death model to try to estimate the relative reproduction numbers of MDR and non-MDR Ural strains. The idea here is that in these trees we have 2 different types of patterns possible. We can have a branching pattern representing sort of the appearance of a new isolate, so that's essentially a birth process, or branches can end, so the death or extinction. And sort of the relative births to deaths represents the reproductive, our estimate of the reproduction number associated with any of the strains. And when she fit her model to these data, again, we lost color here. This is our effective reproductive number estimates for the MDR Urals, quite a bit higher than the non-MDR Urals, which seem to have a reproductive number pretty close to 1. In the interest of time, I'm going to go over this fairly succinctly. So obviously, we have a lot of concerns about what's happening with this Ural strain of multi-drug resistance. There's already a lot of concern about the Beijing MDRs, which we find evidence of as well. But we're focusing our energy right now on these Urals because this hasn't been reported in other places previously. And Ben and Melanie and one of our undergraduate students, Isabel Ranku, who's working with us, have been trying to pull together all of the publicly available TB genomes to try to understand what's happening with Ural lineages across the world. This is unpublished data, sort of very much ongoing work. Here is the tree of all of the Ural strains that we were able to collect, slightly over 5,000 of them. This part of the tree, which looks quite different, are the strains that are most closely related to our Moldovan strains. The outside bars, sort of this darker gray, are the multi-drug resistant strains. This lineage is almost entirely multi-drug resistant. When we unroll this and sort of focus in on this region that we're most concerned about, the other thing that is quite apparent is almost all of these strains are ones that we've sequenced from Moldova. So we don't know what's happening with this. We do see some evidence of it in Europe. That's not surprising given the travel patterns of individuals from Moldova. But what I also suspect is that there's a great deal of underdetection. We just haven't looked hard enough to see where else this might be given its spread in Moldova. Okay. So interim conclusions. Multi-drug resistance in Moldova is concentrated in lineage 2 and lineage 4 and almost exclusively arises through transmission. So well over 90% of our cases of multi-drug resistance we think were infected primarily with strains that were already multi-drug resistant. Clear differences in the spatial distribution and transmission patterns of lineage 2 and lineage 4 MDR-TB, and we have a lot more work to do to understand what the threat of continued spread of these strains, not only in Moldova, but in other parts of Europe, Eastern Europe, and possibly other parts of the world. I'm going to skip this bit. That's Josh, who is a biostatistician who we're working closely with. But just for time, I want to thank our funders, USAID and NIH, and all of our collaborators, especially Valeru Krudu, Nelly, and Alexandru, who welcomed us into Moldova and made it possible for us to do this work. Thank you.
Thank you so much. We have time for questions now, and I'd like to ask everyone to speak into a microphone so that everyone online can follow too.
Hi, thank you so much. Great talk, very interesting. So it's very interesting that you have these two not exactly super related lineages that have this multi-drug resistance patterns, suggesting some convergence there. And I saw that the mutation patterns were actually kind of similar. So the question is, like, evolutionarily, like, do you see that the order in which these mutations appear is the same across the Beijing and Ural ones? Yeah.
Yeah, that's a wonderful question, and I'm just going to pull up, because I think that that this figure shows that pretty well. So what we're seeing here are the different resistance. So genes in which resistance occurs because of mutation. The color of these bars represents the actual codon level mutation. So there are, you can see for some of these, in CAT-G, there are different, that's PNCA. So all of the RPOB and all of the CAT-G mutations are essentially the same. There's a few differences. But that has been well documented both. This is not a new finding. This has been shown experimentally that specific mutations within these genes are sort of low-cost mutations. They allow the strain to retain whatever metabolic activity necessary to do its thing. And I think that's quite interesting. They also seem to occur in a particular order. I didn't show some of the trees in which we try to estimate the years at which these drug resistance mutations seem to sort of pile up on the backgrounds, but there is a very predictable order in which these occur, even though we give these drugs together in combination. Yeah.
Thank you.
This is Barry Blue. May I ask a question? Pronounced
Of course.
Can you hear me?
Yeah.
Ted, first, welcome, and what a wonderful piece of work and presentation. The traditional view in TB, as I understand it, is that transmission occurs from people who have active disease, and people with latent disease, by and large, do not transmit. My understanding is, particularly from studies in Vietnam and islands in the Pacific, household contacts utterly, totally healthy with no symptoms of disease turn out to be major transmitters. So the obvious question with your interesting data, A, have you looked at household contacts of the Ural versus Beijing strain contacts? And is there a greater ability of the 4.1 not to cause disease in contacts, but to be able to transmit infection more effectively?
Barry, great to hear your voice. I'm glad I didn't know you were online, or I would have been more nervous. But it's a fantastic question. So in these data, we can't address that question, but we're in the first year of an R01, where the second aim of that grant, we've collected about 400 cases of secondary TB amongst contacts, most of them household contacts of individuals sequenced in this study, and we're sequencing their strains right now with that exact hypothesis that we may be able to sort of more directly estimate how much sort of secondary disease and transmission is happening from index cases with Beijing or Ural MDR. So yeah, we're totally interested in that.
Great.
Do we have any more questions? not, then let's thank our speaker again.